Hello, everyone. My name is David Mock, and today I will be presenting a project about modeling Chagas disease and some implications where Chagas disease is seen from the point where insecticide spraying is being used to control the amount of vectors present and hopefully cause the number of infections to lower whenever the vectors are killed. So first and foremost, I would like to thank Dr. Aduro for helping me bring together this project and showing me some of the mathematical context, which have been done by him and I believe two other research faculty here at CalU. So I would like to give all the credit to them. And this has essentially just been a really great learning experience for me. All right. So with that, we can ask, what is Chagas disease? Well, there's two stages which are predominant in most Chagas disease classifications. And acute Chagas disease is wherever the person gets first infected and they generally have less severe symptoms, and the chronic conditions are later on after a period of about a couple of months to even further out in their life whenever really severe symptoms become present. And this disease is caused by an infection from the parasite Trypanosoma cruzi. And this is caused whenever kissing bugs or triatoma infestations, they suck the blood from humans and defecate on the skin of the human, where the parasite can then be in contact with that wound and go into the bloodstream and multiply that way. So we can kind of see that here with this image from the CDC. And I'll actually bring up a blown up image here on one of the next slides because it'll be a lot more clear. But to start off, we can see that this bug here, that's the kissing bug. And in this little image, we can see that it has drank the blood from the human and it began to defecate. And a lot of these T. cruzi parasites have been left on the skin and have now traveled into the bloodstream to multiply. And this can be transferred back and forth between the different vectors like animals, between this insect and humans. So it's quite a problem in Central and South America at the moment. So looking at this slide, we can see the T. cruzi parasite. They have quite a unique shape, which is actually known to be characteristic in blood smears, is that the T. cruzi will have this sort of C shape, and it can actually be seen better here. They'll definitely have a C shape, and then the flagellum seems to go behind the organism. And with figure 3, these are just different species of the kissing bugs. I would say the primary one is definitely the triatoma infestations. Most of the research articles for this project have regarded them as the #1 cause for this disease. Here's that blown up image. You can see it probably a little bit better now. This is that kissing bug again. It's on the skin. It drank some of the blood of the human. It's defecated. Now it's multiplying by binary fission. It'll repeat its life cycle like this inside of the bloodstream of the human. Then maybe this bug will go to these animals and then go from those animals to other uninfected people. And that'll essentially be the cause of the spread of this disease. Here is some of the two stages of Chagas disease. And it is quite known that acute Chagas disease, there's varying symptoms. Most people actually don't have very many symptoms at all, and it can be attributed to symptoms that are very similar to viruses like influenza, or even like different versions of the cold, which cause symptoms that are very similar. And generally, it's very hard to categorize when someone is in the acute Chagas disease because they have to take blood smear samples or do a DNA test by, I believe, PCR isolation from the parasite. And in these rural areas within South America and Central America, it's quite expensive. So it's not done very often, unfortunately. And the main characteristic that can be seen within children, and sometimes it happens in adults too, is Romana's sign. That's whenever children or adults have scratched the areas and then they touch their eyes or face, and it causes quite a bit of swelling, as can be seen here. And it's really unfortunate because then those areas can become quite impacted or move from those areas to their bloodstream, which means that they can develop chronic Chagas disease later on. And about 40 to 60% of people are seen to get chronic Chagas disease, and that's where they often have heart infections or gastrointestinal, like esophageal and some stomach or liver infections, where they can have a lot of problems. Generally, it's the most severe tend to be with the heart arrhythmias and cardiac arrests, because those are very hard to stop once those areas are impacted. And then achalasia is whenever the affected person can't swallow food very easily because their esophagus has become so infected by the T. cruzi parasite. Moving on from that, we can see Chagas disease has had many worldwide impacts. One of them being the global cost of Chagas disease is about $7.2 billion at the moment. It's probably a little bit more now because that came from a research article that was a couple years old. And it seems that Chagas disease has more or less begun spreading even more because of certain climate changes and just the growth of human populations over the past couple of years. About 16 to 18 million people have a form of the disease, and this is definitely an estimate because it is less than 10% of the infected human population which has been diagnosed. So they're kind of going off of rates that they've seen over the past, I would say it's been about 40 years that it's been heavily studied and computed mathematically for those type of calculations. And unfortunately, because only 10% of the population is diagnosed, it can spread a lot more rapidly and populations can't be tracked as easily. And that's partly why many mathematical models that Dr. Oduro has worked on have been used to look at things like this, is because it's just very hard to do statistically. And I would like to also say that endemic countries are actually becoming a little bit better with some insecticide spraying, and now there's a lot of concern between just getting rural areas under control with Chagas disease and non-endemic countries getting blood transfusions. It's been known that Europe is being impacted very heavily because they never had to test for Chagas disease in Europe for probably, I would, if I remember the article, it was about 20 years where they never had to test for Chagas, and now they're starting to ramp it up because so many people have migrated from endemic regions who have moved to Europe, and it's becoming quite a problem with the spread there through blood transfusions. Okay. Looking at these images here, we can see the left image that these are areas of a scale where interruption has been caused by like things like insecticide spraying and just generally trying to control insect vectors and trying to make sure that the spread is maintained. It can be seen in northern Brazil and some other countries like Guyana and French Guiana have had quite a problem with not being able to do much in terms of control. And that's just because of resource availability and in general, just not having monetary value and trying to stop such a broad ranging area of the Chagas disease spread. Even Mexico has had problems where it's just such a wide and vast population increase of the vectors that it's almost uncontrollable at this point. Although there is some luck with places in Argentina, and it looks like some portions of Mexico where there is either very low vector type insects which can be interrupted, or it's just more controllable in those areas. The right image shows us that all of the deep orange is all endemic areas, and that's Central and South America. And then the non-endemic areas are in the lighter orange. And in the US, we've actually had 100 to 300,000 cases per year. And that's from multiple reasons, including blood transfusions and just the native habitat is being pushed a little bit because of climate change, and they're having more interactions with humans. All right. So visualizing some of these interactions will allow us to develop a human-oriented model where things like organ transplantation and just blood transfusion are really focused on. And then we also can throw in some other parameters like lab accidents and mother to infant transmissions, and then how these can interrelate with these vectors and how the human can actually pass these two vectors, causing more humans to be affected overall. I believe this next slide to be more mathematical, and this should be fine for now, but I think I'm going to get a drink and I'll be right back. Hello again, everyone. This will be the introduction of the mathematical model. And to start off, this might look a little bit confusing at first, but I'm going to look at this left side here, which tells us the total human population. And the NH to T is going to be our total human population, and that's represented by the susceptible humans, the infected humans that are in the acute stage, and the infected humans that are in the chronic stage. And then these humans and the vectors, usually the insects will be what we are considering the vectors in this case, will be interacting, and this will yield a couple of equations as we can see down here, but I'll actually take us over to this diagram first because we can begin to think about these processes as stages. And if we think about these interacting with each other, we can see that the susceptible vectors and the infected vectors are going to be the main points in which move these infections into humans. And so we can see that infected vectors are going to infect other vectors. So the insects are going to be able to infect insects. And then the insects will be able to infect susceptible humans, which move them to the infected acute stage. And then over a certain period of time, some portion of these will turn into infected chronic stage. So essentially, this is just divided in humans in the top row, and the bottom row is going to be infected vectors. And the way that these equations are essentially built off of each other is we can see that these infected vectors, when they bite a susceptible human and infect them, we can see that that has a certain rate. In this case, we'll make it a parameter because it helps us create an equation that way. And if we look at the susceptible human population, we can see that we're actually subtracting the susceptible humans because that is the amount moving to the infected acute stage. And if we also look at this, we can see that we're subtracting this mu h, that's the natural death of humans. So we have to take account of that. And then the born population of humans, those are all the humans that are born into the population. And we expect that in this model, we're going to say that all born humans are susceptible and not already infected. And if we go to the acute stage in humans, we'll know that some will die from natural causes, not from the disease. And then a portion of these humans will be moved into the infected chronic section. And that's where we see both natural deaths and deaths from the disease, which is denoted by the delta here. And if we also look at some of these other equations, we can notice that this is the migration of susceptible vectors minus this. This is the variability rate of when a vector is going to be put into an infected compartment. And in this case, that's denoted by the tau, the T-looking symbol. And then the essentially the inverse of that is just here because those are all going to be non-infected vectors. And whenever we're looking at this further, we will notice that gamma will play a huge role in this whenever the model is approximated for. And that's because a gamma is the insecticide spraying. So that's going to reduce susceptible and infected vectors, killing the insects and making sure that this infection rate is lessened over a period of time. And essentially this whole model here is going to be put into these equations, thinking about what compartment is being moved over after the interactions take place. So if we think about the importance of gamma and the reproduction number in disease-free equilibrium, essentially what we need to discuss at this point is that disease-free equilibrium is when what we hope whenever we use insecticide spraying will make sure that no humans are infected after a certain period of spraying. And this can be essentially resembled by this RO number. And we know that from the equation, it will be when this RO value is found after you combine all the equations and solve for them, there'll be a resulting number that tells you how many people will be infected over a period of time. And in disease-free equilibrium, we want it to be less than 1 because that ensures that the number of people that are infected will be less than 1 and the disease will eventually fade. But if it's greater than 1, it means that more people will be infected. So especially with viruses going around at the moment, like COVID, there's known that a reproduction number will be around 2 to 5. And that's essentially saying that 2 to 5 people will be affected or infected by the disease whenever one person spreads the disease. And if it's less than 1, that means one person is spreading less than one person will be affecting less than one person, and that means it'll eventually die off. In this case, Chagas disease, whenever in disease-free equilibrium, we will be able to find a value which tells us the amount of insecticide spraying that is needed to lower it below the value of 1. And I believe, let's check the rest of this PowerPoint slide here, make sure I'm not missing anything. I believe that to be everything for this portion of the PowerPoint. And then a brief discussion of endemic equilibrium. Essentially, endemic equilibrium is kind of at the stage that most endemic countries are at now, is whenever there's just so many vectors that it can't be controlled, and there's a point to which the populations remain in equilibrium with each other. So the human population will essentially be a constant, and then the vector population that's infected will be a constant. And essentially those are going to interact and ensure that there's always a disease of in humans in those areas. So Chagas disease essentially will not be eradicated and it'll stay in the population like it is now. And the project goals for this were essentially for me as a student to learn more about how modeling works. And that actually brings me back, if we go back to the slide, whenever we're thinking about this reproduction number in disease-free equilibrium, if we get a certain amount of that insecticide spraying so that it reduces it to that reproduction level of 1 or less, we'll be able to find that as insecticide sprays concentrations are increased, that the susceptible human population will increase, which is actually a good thing. That might sound kind of bad, but it actually ensures that there aren't vectors that are infecting those people, even though they could be infected by, let's say, a smaller population of bugs. But while also reducing that population of infected vectors, we'll be able to obtain a reproduction number which is less than 1. To continue with the idea of my project goals, essentially what I'm trying to do at the moment is approximate some of the values that will be seen in the reproduction number, and then to be able to find some values with an endemic equilibrium as well. So to finish off, my reference page will be here. And this is just the CDC slides, Drugs for Neglected Disease Initiative, and a Scientific Electronic Library online database. And then the research article group members, Dr. Bismarck-Iduro, Dr. Catabo, and Dr. Mialu. So I would like to give credit to all of them and to thank especially Dr. Iduro, because I work directly with him for helping me with this project. So thank you for having me.